I want you to imagine you're walking along the shores of Lake Ontario. You look out into the water and you see something lurking there. Is it the Loch Ness Monster? A freshwater shark? No, it's something much, much worse. It's a harmful algae bloom. So here in Ontario, we have a type of algae called cyanobacteria. This cyanobacteria can produce toxic proteins that can negatively affect humans, their dogs, livestock, and more directly, the fish within our waterway. These blooms tend to form under an ideal set of conditions, being when UV light is high and it's sunny, when water is warm, when water is stagnant, and there's a lot of excess nutrients in the water, such as nitrogen or phosphorus. These nutrients can come from things like agricultural runoff or urban activity and sewage. Now you may be thinking, where are all these parameters met? The Great Lakes in summertime. Lake Erie, for example, has been experiencing a severe harmful algae bloom for the last 12 years. My thesis focuses specifically on how the toxins from these blooms are affecting local fish species here in the Great Lakes. Now, how do we go about determining this? Well, we can take blood from fish non-lethally. So what that means is very similarly to how you may donate blood, we only take enough blood in relationship to the body weight of the fish so that once they're released back into the wild, they won't die from blood loss. From that blood sample, we can then separate it into two components, red blood cells and plasma. That plasma is a liquid goldmine of information for our lab. Plasma contains information on proteins that have been circulating through the entire body of the fish. So for one small sample, we can get information on liver function, cardiac function, neural function, and so much more. We can then compare the plasma protein levels of a fish who was not exposed to the bloom to one who was and see how those toxins are directly affecting biological functions, causing things such as neurotoxicity or even liver hemorrhaging. There's a current gap in the scientific community. There's not enough information on how the toxins from these blooms are affecting local fish species here in the Great Lakes. There's information done on small fish in the laboratory, but not enough on what's going on directly in our backyards. As climate change continues to warm our waters, those ideal set of conditions for a harmful algae bloom to form are going to occur more frequently, and they're going to last longer. My research is not only going to educate the general public and the scientific community on the toxins and the dangers associated with these toxins, but can hopefully lead to future research that can slay these harmful algae bloom monsters once and for all.